My name is Nanthaphong Khamthong from College of Oriental Medicine, Rangsit University. Today I would like to present you our research work in the title of Physicochemical Parameters and Chemical Compositions of Essential Oils from Marjoram and Sweet Basil, cultivated in northern Thailand. So before we start, so let's get to know two herbal plants of our interest in this study. The first one is marjoram. We can call it sweet marjoram. This herb is native to Mediterranean regions, and it has long been used as spices and used in traditional and folklore medicine because it's exhibited various bioactivities such as antimicrobial, anti-inflammatory, and communicative activities. Apart from the use in medicine, the essential oil was used in aromatherapy, and the essential oil contains several bioactive compounds as shown in this slide. The next one is sweet basil. I think it is very well known to everyone here, as we use it in food, commonly used in food, and we can use fresh and dry in food and also in traditional medicine. It helps relieve mental fatigue, cold spasm, nausea, flatulence, something like that. In the international market, the essential oil can be classified into 4 major groups, including European, Reunion, Tropical, and Basil with high percentage of eugenol, which the oil from Thailand belongs to the Reunion Basil oil. So these two herbal plants are among other herbal plants cultivated and promoted in the highland areas of Thailand at the Royal Project stations. They are produced and supply fresh leaf for food and essential oil extraction at the Royal Project. Based on the literature, we found that there is no report on the quality of these two essential oils. So we are interested in the quality study of these oils. So to study the quality of the oils, we focus on the physicochemical properties, including oil yield, specific gravity, refractive index, for example. And after that, we will screen the basic chemical components of the oils using thin layer chromatography, and then analyze the chemical components in the oils by using CAD chromatography and mass spectrometry. All the experiments will be conducted with comparison to the commercial oils that are available in the market. First, we distill the essential oil from marjoram and sweet basil by using water or hydrodistillation for 2 hours. And then we get the corresponding essential oils, and we subjected them to the study. The first one is physicochemical characteristics study. We study oil yield, which we use this calculation to calculate the oil yield. And then we determine the color and odor of the oils by physical observation. For the other physicochemical characteristic includes specific gravity, refractive index. We follow the standard method that described in the USP in the year of 2000. For specific gravity, we use a pygnometer, like a scientific instrument. For the refractive index, we use refractometer. And for the solubility in ethanol, which means the volume of the ethanol, completely dissolving 1 milliliter of the oils. We use three different degrees of ethanol, including 70, 80, and 90%. And for the Tlc analysis to screen the basic chemical of the oil, we use essential oil with comparison to commercial oil from the TCFF or Thai Chinese Fragrance and Flavor Company, and also the standard compounds that are a lot available in our lab. After spotting the sample onto the TLC plate right here, we developed the TLC plate using a mixture of toluene and ethyl acetate. And after we got the developed TLC plate, we visualize the spots of the component by using vanillin sulfuric acid spray. And the last one, Gcma analysis for the determination of chemical component. We use GCMS spectrometer and use the following condition as shown in this slide. We come to the result. This slide summarizes the result of physicochemical properties. So let's start with the first property, which is oil yield. From the result, we found that the marjoram oil, I will call HMO, and the sweet basil oil, I will call HBO. We can see that the HMO yield from our study was comparable to those marjoram oil from Tunisia and Greece, but it was lower than those Egyptian marjoram oil. And for the HBO or sweet basil oil, we found that it was lower than those reported in the literature. And if you want to improve the yield of the essential oil, there are several recommendations to improve the oil yield. Includes seed conditioning, seed treatment with gibberellic acid, organic cultivation with compost manure, or bacterial inoculation. So we can use these recommendations to improve our yield in the future. For the other physicochemical property, color, odor, specific gravity, and refractive index, we found that the oil, the aerodistilled oil from our study, when compared to the commercial oil, they were not much different from each other. For specific gravity of the HMO and HBO, we found that they were in the range of the standard values. You can see that the green letter here. But for the refractive index, the HBO is a bit lower than the standard range. I think it would be due to the different experimental temperature that we conduct in our study. And the last one is solubility in ethanol. We use 3 different degrees of ethanol, and the results show that for the HBO oil, we found that the HBO oil can be soluble in 70% ethanol than the TCFF oil, which was not insoluble. So this can indicate that the HBO contain more polar compounds than TCFF oil. This slide will show you the result of the TLC fingerprint or the TLC analysis of the chemical component present in the oil. You can see in this TLC chromatogram at the lane 4 here, you can see that the HMO oil exhibited about 6 different spots in the TLC chromatogram. And after that, we can propose that at the retention factor, or we call RF, at the RF of 0.78, and 0.56, we can propose that it would be linaloyl acetate or geranyl acetate, and it would be terpenin 4-ol or linalool. For the RF at 0.96, which present in both HMO and TCFF, we cannot propose because it is a minor compound, minor spot, and we did not compare with the standard compound. For sweet basil oil right here on the right side, at the RF of 0.97, which could be detected in both HMO and TCFF, contain mycene, has a minor compound. And we obviously we can see that at the about IF of 0.57 to 0.59, the presence of linalool in HBO can be observed. And for the Gcma analysis, we conduct the experiment in order to identify the chemical component present in the oil. The first one for the marjoram oils. This table summarizes the chemical components present in the oil. We can see that for the HMO, the hydrolyzed marjoram oil, the presence of cyclohexanol with amount of about 39% is a major compound, followed by terpenin and saponin hydrate right here. But for the TCFF oil, the major component is cineol, followed by linalool. The other compound that present in both HMO and TCFF that they are common in both oil are terpenin 4-ol and linaloyl acetate. From the literature, we found that cyclohexanol derivative can be detected in marjoram oil obtained from the USA with about 73%. But the marjoram oil from Egypt, which accounted for 21% of cyclohexanol derivative. So you can see that both oil, HMO, and TCFF, there is a variation in chemical composition. This variation could be due to the different places of cultivation or localities, or different cultivar or species of the plants, plantation environment, and climatic condition, which it would be should be further studied in the future. For the last one is sweet basil oil from the GCMS analysis. We found that in accordance with the TLC chromatogram, we found that linalool was a major compound present in the sweet basil oil, followed by eugenol right here. But for the TCFF oil, you can see that estragon accounted for about 85% or 86% is a major compound present in the TCFF oil. And this also could be due to the different locality, different plant cultivation, because sweet basil from TCFF oil was imported from China. So it could be led to the different in the chemical component. For other common compound that present in both HVO and TCFF were shown in a rectangular text box right here. When we compare to the literature, we found that 5 cultivars of sweet basil from Prague had about 16 to 32% of linalool. But for the oil from Ethiopia, it accounted for about 38% of estragon. So this literature was in accordance with our result. So in summary, the physical-chemical properties of the aerodistilled oil in this study and commercial oils were not much different from each other. Both hydrodistilled marjoram oil and aerodistilled basil oil were different from the commercial sources in terms of chemical composition, which the factor that leads to the variation in the amount of the chemical components of this oil should be further investigated in the future. And also the biological activity of marjoram oil and sweet basil oil obtained from the plants cultivated in the northern of Thailand should be carried out too. And generally, HBO, which is sweet basil oil obtained from the plant in the northern of Thailand, can serve as a rich source of linalool. So I would like to thank you everyone for your kind attention and the funding from the Royal Project and College of Oriental Medicine. Thank you very much, everyone.
Okay, thank you very much. This is very nice presentation. Okay, from the audience, are there any questions from the audience? If you don't have any question, but I have a few questions. Okay, the first one is you mentioned that the level of such as oil or chemical component in oil is vary from the location. Okay, that they cultivate. So what are the factors of the cultivation or the process that have the effect in the oil or the chemical component?
Right. These factors can include different type of the soil that we use, climatic condition, which means the temperature that at the place that we grow the plant, and answer is that culture condition. Some places use synthetic fertilizer. Some places use compost manure or compost fertilizer though. So this factor can lead to the difference in the chemical components of the oil.
From your opinion in Thailand, do you think it is a good enough condition for the cultivation of the two plants? It is good enough in Thailand?
I'm not sure because at the Royal Project they don't control the cultivation condition. They just plant the plants that they obtain from the Royal Project in order to supply fresh leaf, or in order to help the hill tribe people to have a job, to help them to have a job. So generally they don't control the condition that we talk in this presentation.
And the last one, in your result, like TLC result, can you show me the TLC result?
Yes.
Oh no, TLC. Okay. In HMO, okay, in RF 0.96, okay, in 0.96, I think is the intensity of this spot is high intensity. Is the number 2 high intensity. So for your result, you cannot have any control to show that RF 0.96 is what is the chemical, okay, because in your control you don't have the RF factors is 0.96. In my suggestion, you must find some chemical of find some control that have RF factor is nearly or 0.96, because in your result HMO in this RF factor is quite in high intensity. So if you can find this chemical in this RF factor, you can, I think you can develop your work to publish in international journal. Okay, and another thing that I would like to suggest you is about the you should I think you have told me you should find the biochemical activity in biological activity of the HMO and HBO. I think this part is quite important. If you have to find or do in your further work, you can correct your data and submit in the high impact of the journal.
So that would add up the value, the quality of the paper. Thank you very much, ครับอาจารย์
Thank you very much. Thank you very much for your presentation.